I hope you understood what I talked about, what I taught yesterday about the genetic terminology, some of the words. And furthermore, we have to go further. Number one, again, the phenotype. What is phenotype? The external visible appearance of an individual. External visible appearance of an individual as per the book, observable characteristic of an organism. This is the definition as per the book. So the visible appearance or observable characteristic of an organism is called phenotype. Suppose you are taking, for example, so I am taking phenotype as well as a genotype together. Genotype. For example, tau. We have two genotypes. The tau may be homozygous or heterozygous. This is one. And we are taking another one, the recessive character, dwarf. What is the genotype? That is what we have the recessive gene in homozygous condition. So for tau, that is capital T or capital T small t. So now the factor responsible for the expression of a dominant character just represented by capital T, and the alternative form is just actually that is recessive one, that is alternative letter. So here you can find the gene responsible for the dominant character is called dominant factor. The gene responsible for recessive character is called recessive factor. Here capital T is the dominant factor and small t is the recessive factor or gene. Now the dominant factor can be expressed both in homozygous and heterozygous conditions, whereas the dwarf character or the recessive character can be expressed only in homozygous condition. So the dominant character is expressed both in homozygous and heterozygous condition, whereas the dwarf character is expressed only in homozygous condition. That is about the nature of the factor, dominant factor, then recessive factor, dominant individual, recessive individual, or dominant trait and recessive trait. This is another what is called terminology. It's a point where a particular gene is located. This is another terminology. For example, this is the fifth locus. Say an example, the fifth locus, that means the fifth point where the fifth gene is located. See, that is called as a locus. The point where a particular gene is located is called actually, that is locus. Mendel proposed two rules while conducting experiments on garden pea plants for monohybrid cross. These two rules now called as laws of Mendel. So these two rules, rule number one, that is also the law number one, law number two, that is based on monohybrid cross. What is the first law? Law of dominance. This is the first law. The first law, law of dominance. Number two, law of segregation. Law of segregation, or law of purity of gametes. Law of purity of gametes. These two rules were based on monohybrid cross. So based on the observations of monohybrid cross, he proposed these two rules, what are called the laws later, law of dominance, law of segregation, or law of purity of gametes. These two laws played a major role in the history of evolution. Now let's take what is law of dominance. When two individuals which are pure for contrasting characters are crossed, only one of the characters appear in F1 generation. The character which appeared in the F1 generation is called as a dominant character. The other character which is not appeared, which is masked is called as a recessive character. This is the basis for law of dominance. You know that one, he studied contrasting characters. The contrasting characters are controlled by units of entity, discrete units called actually the factors. The factors always occur in pairs. Now, law of dominance normally gives explanation to the monohybrid class. What explanation it has given to monohybrid class? So, the factors occur in pairs. For example, if you are taking just actually tall plant and then low plant, they are crossed. These are the parallel combinations. We have received the F1 generation that resulted also tall. So this tall is a hybrid tall, and this is what's called the pure breeding variety. Now in the F1 generation you can see only one of the factors appear in the F1 generation, and both the factors appear in the F2 generation. So it explains or it gives explanation to the monohybrid class. What is that one? Number one, the recessive factor, or what we call the recessive character, the trait, appeared only in the F2 generation and reappear in the F2. So that's a meaning. So I repeat once again. Now the recessive factor or the recessive trait appeared only in the F2 generation, whereas both the recessive and the dominant traits, the recessive and dominant factors appeared in the F2 generation. That is the explanation given by this law of dominance. So only the recessive character appears in the F2 generation. Actually, both recessive and dominant characters appear in the F2 generation. That means one of the characters, namely the dwarf character, is suppressed or masked in the F1 generation and reappeared in the F2 generation. So it also explains the proportion of 3:1 ratio obtained in monohybrid cross in F2 generation. So what is the importance of law of dominance? It actually explains the law based on the monohybrid cross, or it gives explanation to the monohybrid cross. I'll repeat. Now, appearance of only one character in the F1 generation, that is number one, that is the tall character. And appearance of both the traits in F2 generation, that is the second one, that is a proposal according to the law of dominance. It also proposed the appearance of, that is 3:1 ratio, or it also proposed the proportion of 3:1 obtained in F2 generation. So this is all the importance of law of dominance. Appearance of only one character in F1 generation, appearance of both in F2 generation. It also explains the proportion of 3:1 ratio obtained in F2 generation. So again, what is law of dominance? When two individuals which are pure for contrasting characters of a pair or cross, only one character appears in the F1 generation, and that is called as a dominant character, and the gene is called dominant factor. The one which is not seen in the F1 generation is called as a recessive factor. That is also a recessive character. Either we can say recessive character or recessive factor. With reference to factor, we can say recessive factor. With reference to character, we can say what is called recessive character. So this is called law of dominance. It is based on the results of monohybrid cross. Law of Mendel, what is called the law of dominance, is based on the monohybrid cross. Now the second law of Mendel is called law of segregation, or law of purity of gametes. It is also based on the observations of monohybrid cross. What is the law of segregation? Once again, we are crossing two individuals which are pure breeders. I am taking tall pure breed and dwarf pure breed. So this is pure breed and that one is also pure breed. These are the parents. When they are crossed, we are getting F1 generation resulting in what is called hybrid tall. All the plants are tall but hybrid. So this is hybrid tall, this is pure tall. Now in this case, so when you are taking F1 generation, we are getting hybrid tall. When two hybrid talls are crossed, when two hybrid tones are crossed, we are getting the F2 generation. We are getting the F2 generation in the ratio of 3 tone and 1 dwarf. 3 tone, 1 dwarf. We'll study about this one later in monohybrid class. So we are crossing two pure breeding varieties for contrasting characters. Then we are getting F1 generation, the hybrid tone. Then two hybrid tones are self-fertilized or self-pollinated. This is what is called the cross-pollination or cross-fertilization. And this is called self-pollination or self-fertilization. That resulted in F2 generation with a 3 tone and 1 dwarf. Now, what is law of segregation? So, if we are taking the capital T and small t in F1 individual, there is no blending of these two alleles or genes. There is no blending. Blending means there is no mixing. So both the characters are seen as such in the F2 generation. See, both the characters appear in the F2 generation. That showed that the two genes remain together without mixing or without blending. The appearance of dwarf character once again, that is in F2 generation, indicated that there is no blending or mixing. Both the characters appear as such in the F2 generation. Although one of the characters, namely the recessive character, is suppressed or masked in the F1 generation. So there is no appearance of this recessive character in F1, but appearance of that recessive character in F2. And this is the analysis for what is called law of segregation. What's the meaning for that one? So there is no mixing, there is no blending. During gamete formation, the two alleles of a pair, they segregate and separate, so that each gamete receives only one allele of that pair. This is only one gene of that pair. This is called law of segregation. The meaning for the word separation. The two alleles, the dissimilar alleles, what we have in F1, just normally the two alleles in F1 remain together without mixing. They segregate and separate during gamete formation, and so that each gamete receives only one of the two alleles. So this is called law of segregation. As normally the gametes contain only one of the two alleles, the gametes are always pure, they never hybrid. The gametes are always pure, having only one allele, they never hybrid. Hence this law is also called law of purity of gametes. Law of segregation, segregation and separation of the two alleles, entering the two alleles separately into two different gametes. And each allele has either capital T or small t. Hence called law of purity of gametes. That means the gametes never hybrid, they are always pure, having only one allele for a particular trait. Suppose we are taking homozygous individuals. This is homozygous. We are taking homozygous individuals. They produce similar gametes. They produce similar gametes. I am taking another example of homozygous individual, recessive homozygous. Here also we have received gametes, and all the gametes contain only small t or recessive allele. So this is also what we have, homozygous individuals produce similar gametes. If we are taking heterozygous individuals, these are all heterozygous individuals, they produce dissimilar gametes in equal proportion. They produce dissimilar gametes in equal proportion. That is 50% of the gametes with a capital T and 50% of the gametes with a small t. So the appearance or the reappearance of dwarf character in the F2 generation indicated that there is no mixing or blending of what is called the alleles of the genes. During gamete formation, the two alleles separate and enter into separate gametes. So each gamete receives only one of the two alleles, not both. That's why I say the gametes are always pure. Hence the law is called law of purity of gametes. So this is about what we call the law of purity of gametes. The first law already we discussed, law of Mendel, that is law of dominance. Involving a single character inheritance of single character, we can say crosses involving the inheritance of single character is called monohybrid cross or called monohybrid crosses. So it involves the inheritance of a pair of alleles of a single gene. Involves the inheritance of a pair of alleles of a single gene. Suppose you are taking the parents. This is the illustration what we have. So we have pure breeding tall plants. This is a phenotype, and pure breeding dwarf plant. So these were the plants taken for artificial cross or we can say that is cross pollination, because we are taking two different traits of a carrot. This is the phenotype. Now these are all the pure breeds. Now this is the genotype. Both the genes are similar, hence called as homozygous tall, and that one is called homozygous dwarf. So this is the dominant character, that is a recessive character after the results obtained. The letter was given to the dominant gene after the results obtained in F1 generation. Now what are the gametes produced? What are the gametes produced? So as it is a pure breeding plant, all the gametes produced by the tall plant is similar. Let us take for example the tall plant. I am taking that is a male, that is a pollen grains. Then we are taking also the dwarf, pistil. I am taking in such a manner. Now the gametes produced, all the gametes produced by the tall parent, the pollen grains contain capital T, that is a dominant factor. Now the dwarf parent produces gametes, and all the gametes with a factor that is recessive factor. Now the cross, the fertilization occurs, the gametic union occurs, leading to the formation of the F1 generation. We are getting F1 generation. Now, as per the law of segregation, the two alleles separate and enter into each gamete, and the union of these gametes at random produces a diploid individual that is called the tau. This is hybrid tau. This is hybrid tau. This is hybrid tau. Now this is what's called the cross pollination, the results obtained in F1, that is hybrid tau. As the tau is appeared in the F1 generation, it is considered as a dominant character. That's why we are using the letter capital T. Another character, what is called the dwarf character, is not appeared, not seen. It is being masked, hidden. The F1 generation is called as a recessive factor, and the character is called recessive character. Now, if you are getting what is called the F2 generation by self-pollination, so that is called self-fertilization. Self-fertilization or self-pollination of F1 hybrids. So the non-two breeding or two breeding are called heterozygous. Non-two breeding, the Mendelian non-two breeding, what is called pure breeding individuals are heterozygous, which are called hybrids. So this is heterozygous. Now we have actually this process, self-fertilization of F1 hybrids. This is cross-fertilization. We are taking two hybrid tons. We are taking two hybrid tons. Mendel did the cross between two hybrid tons. Hence it is called self-fertilization process or self-pollination process. Now we are taking two hybrid tons or two heterozygous tons. Now what are the gametes produced? What are the gametes produced? Now each individual produces two different types of gametes in equal proportions. 50% of the gametes with the capital T and 50% of the gametes with the small t. This is because the two alleles in a hybrid do not mix, do not blend. There is no mixing, there is no blending. That's why the two alleles get separated as such without mixing. Here also we have just actually two different types of gametes produced, 50% to each. Now what will happen? Now we have two different types of gametes on either side. Two different types of gametes on either side. So the probability and possibility of individuals would be 2 into 2, 4. The total number of individuals produced is equal to, that is the simple multiplication, the random union of the two gametes. As we have two different gametes on either side, we are getting actually totally 4 individuals. That means each gamete has a chance of fertilizing this one and that one. And similarly, taking this gamete, it also fertilizes this one and that, because of random union. So we can just represent the products of this self-fertilization process in F2 either by means of four-line method or by means of what's called Punnett square. So as a result of actually the fusion of the gametes by random union during fertilization, we can find out the phenotypes and the genotypes of the progeny by means of a diagram, a graphical representation, and that one is called Punnett square. That one is called Punnett square. So we can represent the products of F2 either in the form of Punnett square or in the form of four-line method. Four-line method. So the phenotypes and the genotypes of the progeny of F2 resulted due to the union of gametes during fertilization can be explained clearly by means of a Punnett square or four-line method. The Punnett square was actually represented by a person, a British geneticist, Reginald C. Punnett. He was the first person just actually proposed this Punnett square. So what is Punnett square? Now this is a graphical representation to calculate the genotypes, actually the possibility and the probability of the genotypes in the progeny with reference to particular trait. So what is actually Punnett square? It is a graphical representation to calculate the genotypes. You can calculate the probability and possibility of genotypes of the offspring, and that is called, that is Punnett square board. So what is actually Punnett square? This is the definition. So it is a graphical representation to calculate the possibility and the probability of genotypes of the offspring. Okay, by which one can find out, that is the genotypes and phenotypes. And I will take first one, that is a four-line method. I am taking this gamete, fertilizing this one. Maybe this is what is called the male and this is female. So the pollen grains are dusted on the stigma. Now this is one. We are getting this one variety. It also has a chance of equally fertilizing this one. We are getting this. We are taking this gamete. It has a chance of fertilizing this one. It also has a chance of fertilizing this one. So this is what is called the four-line method. This is called what is known as four-line method. Now in the four-line method, at the end of actually F2, the results obtained were 3 tall and 1 dwarf. 3 tall, 1 dwarf. And this is the phenotype. This is the phenotype. Okay, or we can represent what is called the first genotype, then the phenotype. So the phenotypic ratio would be 3:1. If we are taking the genotypic ratio, not all the 3 talls are same. They are different in their genetic constitution. One of them is having what is called homozygous genotype. The other two have heterozygous genotype. So, if we are taking the genotype, one individual with a homozygous genotype, 2 individuals with a heterozygous genotype, and 1 with a homozygous recessive genotype. So the genotypic ratio would be 1:2:1. So in F2 generating the following results obtained. Totally, he obtained in terms of numbers 1064, 1064 individuals So out of which 787 tall, 277 that is growth. He made these numbers into definite ratio, that is 3:1. So in original condition, he received 1,064 individuals, of which 787 individuals were torn. The remaining 277 were dropped. He made into a definite proportion, that is 3:1, that is a phenotypic ratio. Now this is the four-line method to represent what we have, that is the result of F2. Now I will take what is called the Punnett square. The results of the progeny with reference to the phenotypes and genotypes well represented or can be clearly understood with the help of Punnett square. Now I am taking the male gametes on one side and female gametes in another side. You can represent this one either in the form of square or in the form of parallelogram. Now it is a simple probability, a simple multiplication. So what is Punnett square? I mentioned about the board. It's a graphical representation to calculate the probability and possibility of the genotypes in the offspring. Now the probability, you know that one, it has the ability of fertilizing. Now this is the sperm. The sperm has the ability, here it is the pollen grain. The pollen grain with the capital T, the dominant factor, can fertilize what is called, that is the egg or ova or the ovule in the stigma to form, that is homozygous stalk. It may also have, actually if you are taking another one, it is being fertilized by a gamete with recessive gene that resulted in tall, but this tall is heterozygous. We are taking another female gamete, or we have, that is in the plant, that is ovule. So it is being fertilized by, that is a pollen with the dominant factor that results in tall. It is also heterozygous. And it is also being fertilized equally at random. This is a random domain only by gamete, the male gamete with the recessive factor, so that we are getting what is called the dwarf. So out of the three talls, we have only one is homozygous tall. The remaining two in the diagonal line, these two are heterozygous tall. Only one is homozygous tall. The remaining two are heterozygous talls, or we can represent in the form of what is called a triangle. So these three are the talls. Out of these three talls, only one is homozygous tall, and now in this diagonal line we have heterozygous tall. Then only one dwarf individual, that is homozygous recessive. We know that one, the recessive character is always expressed in homozygous condition. So the genotype obtained in F2, one homozygous tall, two heterozygous tall, and one homozygous recessive. Then the genotypic ratio is 1:2:1. What is the external visible appearance? So out of the four individuals, the probability, the possibility of the phenotypes are 3 tall and 1 dwarf. So the phenotypic ratio based on the external visible appearance, or we can say that is the observable characters of an individual, the ratio 3:1. So the appearance of dwarf character, once again, this is the reappearance of dwarf character once again in the F2 generation, indicated that the two genes capital T and small t, these two genes do not mix with one another, there is no blending. That is why both the characters are seen as such in F2 generation, though one character is not seen in the F1 generation. So the reappearance of actually the dwarf character only, there is a conclusion, and that is what is called the result, that gives a proof that there is no mixing of capital T and small t, that is called blending. This is called blending. There is no blending. There is no mixing. That's why we have the reappearance of once again the dwarf character in the F2 generation. in F2 generation. That means we have two contrasting characters, that is tall and dwarf in the case of garden pea plant. We study simply the genes are responsible for the development of such characters. What is the basis for that is the development of such tall and dwarf characters? For that you have to know the functions of the gene at the molecular level, as we have seen in the case of purple and white flower. Now, molecular characterization of Mendel's gene in plant height. That is, what is the role of genes? How far the genes are responsible for? That is, the expression of such characters. That is, the molecular explanation of tall and dwarf traits. The molecular explanation means at the gene level, how far the genes are functioning, nothing but the DNA level. Now we have tall plants and dwarf plants. Now, the pea plant cells have the ability of synthesizing a hormone gibberellin. This is a hormone, gibberellin. This is concerned with, you know that one, the length of the plant, the height of the plant. When the plants are actually supplied with the gibberellins artificially, resulting in stem elongation, otherwise called bolting process, you studied in 11th standard and physiology. Now, the gibberellin actually found in a precursor molecule, it's being converted into an active gibberellin molecule, what is called GA1. This is an active gibberellin molecule. Now, all the cells have the ability of converting this precursor, means the forerunner, that is the basic molecule, into active gibberellins. In the case of tall plant, it is being brought about by an enzyme involved in what is called gibberellin synthesis pathway. This enzyme is coded by the gene, that is LE gene. We have already seen the gene for height. Now, the gene, at least one gene, is responsible. We have actually homozygous condition and a heterozygous condition. At least one or two dominant genes responsible for the expression of what is called the enzyme, which is involved in biosynthetic pathway, that is gibberellin synthesis pathway. Now the gene or allele one dominant letter, that one codes for the functional enzyme. So when dominant gene is present, it expresses itself and produces the functional enzyme, so that the conversion of gibberellins into active gibberellins taking place. That results in the elongation of the stem, so that the plant becomes tall. Now what about in the case of dwarf plant? So the tall plant is a dominant character. The gene for the expression of the enzyme is also dominant. Now this is what is called the genotype of that is tall plant enzyme responsible for conversion, or it may be in heterozygous condition. So these are the two genotypes responsible for the expression of the enzyme which converts gibberellins into active gibberellins. So this is what about the dominant character, the tall plant. In the case of dwarf plant, now the gene is recessive. So dwarf plant is homozygous recessive, you know that one. The gene for the production of the enzyme to convert the gibberellin's precursor molecule into active gibberellin is under the control of dominant gene. But here there is no dominant gene is present and both the genes are recessive. So there is no formation of enzyme, no enzyme production. And as such, there is no conversion of gibberellin's precursor molecule into active gibberellin GA1. Now the allele, that is recessive allele LE, that codes for non-functional enzyme. So there is no functional enzyme produced. This is because of the presence of recessive allele and the absence of dominant allele. So there is no growth of the plant due to the absence of gibberellin hormone responsible for the growth of the plant, increase in the length of the plant. There is no increase in the length of the plant. As such, the plant is dwarf in nature because of the absence of what is called enzyme, which converts inactive gibberellins into active. And this enzyme is coded by a gene. The gene is in recessive form. So the allele codes for only non-functional enzyme. That resulted no growth, so the plant is doomed. So this is the molecular basis or the functional basis of gene. Either the presence of tall plant or the dwarf plant in the case of